Hello, good afternoon, everyone. Welcome to Restore America's Estuaries webinar series. My name is Hillary Stevens. I'm the Coastal Resilience Senior Manager here at RAE, and we are very happy to welcome two speakers today coming to us from Galveston, Texas, to talk about living shorelines. Our presentation topic today is evidence for small-scale living shoreline viability in Galveston, Texas. In case you're not familiar, Restore America's Estuaries is a national nonprofit. We are a member of an alliance group of 10 member organizations that you see on the map here located around the country. We serve as an advocate for these organizations in Washington, D.C. and a convener. We host workshops and conferences on a variety of topics. We also administer a number of grant programs to fund restoration efforts. Among our events that we host is the RAE Coastal and Estuarine Summit. Our next summit will be in New Orleans in December, the 4th through the 8th. We are very excited and busily getting ready for this. The deadline has passed for proposals for oral presentations, but we are still accepting proposals for posters. So please consider submitting to let us hear about your research or the project that you're working on, and we hope to see you in New Orleans in December. We also have a number of different things going on. Registration is open for the summit in the next couple of weeks. In mid-July, we will be celebrating NOAA Habitat Month with a webinar featuring the Teddy Roosevelt Conservation Partnership, Ducks Unlimited. So please stay tuned and you'll get more information about that. Again, that'll be July 19th. If you are interested in hearing more about our upcoming events, please look for us all over social media where you'll get the latest information. And without further ado, let me introduce our speakers today. We will be hearing from Jim Daberstein, who is on the faculty of Lee College. He teaches environmental science at Lee College in Baytown, Texas, and is engaged in grant-funded ecosystem studies in the Galveston Bay estuary with his students. These include gathering data to be used for adaptive management of ecosystem restoration in aquatic habitats, comparing the functional aspects of biologic communities across large-scale estuarine habitat restoration designs, and studying the biologic community characteristics associated with small-scale shoreline restoration in comparison to natural reference marshes and armored shorelines. Our other speaker today is Haley Leha, who is the Habitat Restoration Manager at the Galveston Bay Foundation, which is one of our member groups. She has a BS in Ocean and Coastal Resources from Texas A&M at Galveston, and an MS in Coastal Zone Management from Nova Southeast University. She has over 10 years of experience in natural resource management and habitat restoration. Haley has worked with the Galveston Bay Foundation since 2015 and currently serves as the Habitat Restoration Manager. She oversees the oyster shell recycling program and manages the foundation's oyster reef restoration efforts, as well as Living Shorelines programs. All of our attendees are muted today, as this is a webinar format. If you have any questions, please feel free to enter them in the Q&A, which is at the bottom of your screen, and I will take those at the end and read them out to our presenters. So with that, let me hand it over to you, Haley.
All right, just give me a moment while I share my screen here.
That looks good.
Excellent. Progress forward. There we go. Can you hear me okay, Hillary?
Yes,
Excellent.
You look fine. Go right ahead.
Okay, well, thank you for that great introduction, Hillary. It's great to be on this webinar, such an honor. As Hillary mentioned, we are a RAE member group at the Galveston Bay Foundation. And so I've been working with Jim on a project for many years now to assess some of our living shoreline efforts here in Galveston Bay. So I'm going to just give a brief background introduction on what we do here in our region in terms of living shorelines, where we work, how we implement those projects, and then I'll hand it over to Jim to get into more of the research side of things and some of the results he's come across. So I always like to start with where we are, just to get everybody oriented. Of course, we're in the Galveston Bay region, and the foundation, I always like to call this out because our name can be a bit of a misnomer. The Galveston Bay Foundation actually works all around the Bay system, not just on Galveston Island. So we actually work on land in all 4 counties surrounding that system, as well in the waters of that entire estuary. So working in central Galveston Bay, as well as all of those sub-bays. And one of the reasons I wanted to point that out is because the work Jim has been doing actually looks at a wide range of the system itself. We have sites up in Trinity Bay, Central Bay, and all the way down into West Bay. So it really captures that breadth of that huge estuary here. Now, being such a large body of water, we're of course subject to many erosive forces. I'm sure you guys know we're located near the Houston Metroplex, and we are home to the Port of Houston. So we get pretty severe amounts of ship traffic and the resulting ship wake actions, particularly along that western shoreline of the Galveston Bay system. So that results in a lot of erosion along our shorelines. We're also exposed during the winter months with our northern fronts that essentially blow out the water from the bay. And of course, the prevailing southeast winds also wreak havoc on our shorelines. And that's coupled with subsidence, pretty much historical subsidence, particularly on the northern reaches of the bay. We have some areas that have lost up to 8 feet due to subsidence. And then if you notice in Trinity and central Galveston Bay, those larger bodies of water, that huge fetch really does impact some of our shoreline erosion and stability as well. So in conjunction with those issues, our estuary is also faced with some severe habitat degradation. Our estuary program here locally has actually named it as one of the most critical issues facing our estuary. We've lost tens of thousands of acres of both fresh and salt marsh, almost 2,000 acres of seagrass, and nearly 60% of our oyster reefs. And I mention this because these are some of the reasons the Galveston Bay Foundation has focused on living shorelines as a way to not only protect our important preserves and properties, but also to help restore some of this habitat. And it's just one of the methods that we use to do this. So to date, Galveston Bay Foundation has protected over 20 miles of shoreline. And as a result, about 50 acres of marsh have been restored behind those wave barriers. Now, that's also not to mention that those wave barriers do create oyster and mussel substrates. So we do get some additional reef restoration in conjunction with some of these living shoreline projects as well. So before I dive too far into how we implement our living shorelines, just want to make sure everybody's on the same page with what we actually define a living shoreline as. So in Texas, we simply align this with a shoreline management option. It's a way to work with nature to both restore, create, and protect while also allowing some protection for your shoreline. So it's literally just an alternative to bulkheading or using some kind of a revetment, some kind of that traditional armoring, and allowing those natural processes to take place. So you can see some photos up here. This is of our Shipe Woods Preserve up on Trinity Bay. This is a pretty common example of how we implement shorelines here in Galveston Bay. And I always like to show this cross section. An engineering firm here in Texas put this together. I think it's a great example of how we typically implement these projects here along the Texas coast. If needed, we'll install some kind of a wave barrier offshore, whether that's rock or something lower profile like oyster reef. That of course slows that wind and wave energy down, allows sediment to fall out of solution behind there. And then we'll come in and plant with the native marsh species, which locally here is Spartina alterniflora. And then if needed, we'll actually install upland or coastal prairie species as well, just to further stabilize that sediment. Now, of course, this is what we are trying to prevent or stop. The photo on the left there is actually at the foundation's Kemah property, right on that open side of Galveston Bay I mentioned that gets hit very severely with ship wake action. And so this is a prime example of what happens when a bulkhead fails. The site once had a bulkhead about 40 feet offshore. It failed, and this is what we were left with when we purchased that property. But good news, we've now installed a living shoreline and it's looking great. We've got some great marsh establishing. And then I wanted to show this other photo because this is another prime example of what we see happening along the Trinity Bay shoreline and some other areas that receive some pretty intense erosion. You end up getting a lot of scouring and kind of this bluff or cliff face here that then you literally just have chunks of land falling off of it. So of course, these are some of the options that folks use to armor their shorelines in the past, very traditional approaches using riprap of some sort, just to line right on the shoreline there, a type of revetment. And then of course the bulkheads. But in Texas, most of these only have a lifespan of anywhere from 15 to 25 years, depending on how many storm events they're hit with, how well they're built, that sort of thing. But in the end, they end up causing even more severe problems. Particularly that picture in the upper right-hand corner there, you can see what a mess is left behind after that bulkhead fails. It had already started eroding before it fully failed, and then once it gets hit with that final blow that causes its destruction, you end up with even more severe erosion afterwards. So as an alternative, these are the kind of living shoreline projects we are working to install, not only adjacent to some of our preserves, but also working with landowners to find this as an option for their private properties as well, to give them that long-term resilience that can survive storm events versus these bulkheads that are only going to last 15 to 25 years. So in Galveston Bay in particular, these are some options that we look at for addressing shoreline erosion and also incorporating the natural habitat. Obviously, it goes without being said that cost is a huge factor here. So I like to plot this out just to make it clear. We're looking at erosive forces and cost, and these end up being a lot of deciding factors on how we implement the projects in our region. Of course, when you're looking at a landscape view, if you're in an area with less intense energy, the lower energy spectrum, sometimes we can just simply plant. We're able to plant that intertidal native species, maybe even some on the upland slope going from low to high into coastal prairie to help secure that sediment. When you get into areas with a little bit more energy, we'll oftentimes install what we like to call a low profile breakwater. Essentially, that's any kind of structure that can provide some wave attenuation. Oftentimes that'll look like reef domes, bags of recycled oyster shells, or even sackcrete bags like you see in the bottom right-hand corner there. And those are also very valuable because they create excellent substrate for oyster growth and recruitment. And then of course, at the higher spectrum of erosive forces, like on those areas of the open bay, we oftentimes are installing these larger rock breakwaters, what we like to call our high profile breakwaters. And that just simply means it's going to be a structure that's a bit taller to withstand some of those more severe storm impacts. And I also wanted to add in, the Galveston Bay Foundation is very fortunate, and we've been able to implement so many living shorelines here in Galveston Bay thanks to our eco center. It's a nursery where we grow out our native smooth cordgrass, the Spartina alterniflora. And with that, we're able to actually take native seed, grow it out in our nursery, and then transplant to these project sites, not only for living shorelines, but other marsh restoration efforts. And all of this planting, or at least 95% of it, is done by volunteers. So just a quick shout out to our awesome army of volunteers that have made a lot of these projects possible. And to date, the foundation has installed over 30 living shorelines. About half of those have been in association with a private landowner. And so prior to my time at the foundation, my predecessors, actually like Jim, that I'll be speaking here in a moment, and Leanne Wilde, they're responsible for a lot of these landowner partnerships. And I bring that up because I wanted to give you a brief history of where we've been in implementing these living shorelines with landowners. And this is so incredibly important, especially here in Texas, because the majority of our waterfront property is privately owned. And while they may have a small impact on 100 feet of shoreline, you add that up and that's the majority of our coastline. So we're really trying to target some of these landowners, even individual corporations, to work with us on installing this more eco-friendly, more resilient coastal protection. So in the past, as I mentioned, some of the projects Jim has worked on and my predecessor, Leanne, they were actually able to secure some funding through U.S. Fish and Wildlife Service, as well as a state program called CAP, to do a cost share approach with a few of these landowners. And of course, that provides a huge incentive for these folks to go ahead and implement this more eco-friendly project along their shoreline. But now, fast forward to present day, funders are wanting larger impacts. When you install a living shoreline adjacent to private property, you maybe get a fraction of an acre of marsh restoration. And there's also some objections from some of our funding partners to support these private projects. Even though the living shorelines are being installed adjacent to the private property, there's still a little bit of concern there to allow federal dollars or state dollars to support that. Now, the big issue here that we're facing in Texas is reduced landowner interest in living shorelines, mainly because the cost is high, but also because the time frame is so incredibly long for these folks to implement this project. A lot of the individuals I talk to, they're gung-ho, ready to do something more eco-friendly. They appreciate the fisheries benefits and the long-term resiliency of a living shoreline. But then we start talking about permitting. Unfortunately, we have a very long time frame for getting all of those permits completed. Typically, it takes up to 2 years to get our state permit that allows for any construction on submerged lands, because those are actually owned by the state, specifically the General Land Office. And that's just a very lengthy permit process. Even for us, the foundation, our projects, we have to go through this, and that's typically the holdup. So, we have landowners who come to us who are ready to go, but then we give them this time frame and it's just not doable. They're watching their land literally fall into the bay. And on the contrary, they can go out and get a bulkhead ready to go within a month's time and installed for much cheaper. So it's very hard to dangle a carrot for living shorelines when they've got this quick and easy alternative. So that being said, at this point, the Galveston Bay Foundation is really serving more as a resource hub and an outreach partner. So we do provide consultation for landowners. We'll actually go out and do what we're calling a pro bono site visit and give them some guidance on what are some of the next steps they can take if they do want to pursue a living shoreline. And then we're also working with a variety of partners, both university partners and agency folks, to help find a way or a resolution of making this more approachable for landowners and at least educating them. So we're coming up with a few tools. You can see here, we've got an online map viewer that allows individuals to zoom into their little segment of shoreline, click on that segment, and it provides more eco-friendly recommendations for their shoreline protection. So what's next? Obviously, we need to find some incentives for landowners, whether that be funding, changes in the regulations, maybe some tax breaks. But bottom line, we need documentation that shows just how resilient and beneficial living shorelines are, particularly in comparison to the traditional bulkheading. And so that's where Jim's research comes in and is so incredibly important to our work. So before I hand it over to Jim, just a few slides on his sites that he's been studying. So he's been looking at 3 different living shoreline projects. These were all done under the umbrella of Galveston Bay Foundation. And as I mentioned, they kind of capture the whole length of the bay system. So starting up north, the Sullivan Living Shoreline, that is adjacent to a private landowner's property. The Clear Lake Forest Park Living Shoreline, that's actually adjacent to a public park. And then last but not least, the O'Donnell Living Shoreline near West Galveston Bay, that's another private landowner there. So this is a drone image of the Sullivan Living Shoreline. You can see the 2 phases there that were installed in 2011 and 2013. And at this site, we utilized a high profile breakwater approach using rock, and then of course, marsh planting. Lots of student volunteers made that possible. The top photos here show that site before the implementation of that living shoreline, and then the bottom are after, just recently taken in the last, 2 years, I want to say. You can really see how that shoreline has even started to naturally grade down to a smoother slope, and how well that marsh grass has really taken hold. And then moving down to Central Bay, the Clear Lake Forest Park. This site is along a calmer bayou, so we were able to install a little bit lower profile rock breakwater. Now, we did have to bring in some fill material at this site to bring the elevation up to the right level for the marsh grass to grow. But you can see now how we have a really nice succession from low to high marsh here. And these are some aerial images of that site, just showing before and after construction. All right, and then moving down into West Bay, again, as I mentioned, this is a private landowner's property. They installed sackcrete bags, so basically marine grade concrete along their shoreline, just to give the marsh a little added protection and to create some oyster habitat. And then of course, there was some marsh planting done to really amp that up. And this is the land view of that same site. Actually, those are some of Jim's students out there getting their samples, I want to say last spring. So with that, I will hand it over to Jim.
It's the second time I've done that. I have to unmute. Um, so um, I'm glad to be here, and thank you to Haley and all of our partners. This has been a very long-term, as Haley pointed out, a long-term partnership, primarily between Lee College, the University of Houston Clear Lake, and the Galveston Bay Foundation. But we have other partners that have provided funding, technical expertise, you name it. There's a lot of words on this slide, and my intention is not for you to read all of those, but what I wanted to build on is a couple of things. One of which is the time frame that it takes to really implement something like this. The sites that we're looking at are somewhat unique compared to what I see referred to as living shorelines in the literature, and certainly among some conference presentations of that, which in many cases can be very large. In this particular case, as it relates to the Galveston Bay Foundation program, a lot of our sites, especially those that are working with private landowners, are very small. In some instances, we may only have 50 to 75 feet of shoreline to work with. So this can be, again, a challenge in terms of scale. And it also can be a challenge in terms of sampling. Sites have a lot of snags. They have a lot of issues that are particular to the type of sites that they are. So in many instances, we had to develop methods that would allow us to actually gather some data for these sites. So that process started some time ago on a larger project that we implemented in the lower part of Galveston Bay, doing a comparison among different restoration approaches. It was also a very shallow embayment, again, because this area has suffered from subsidence over a large swath of the Galveston Bay area. A lot of former uplands, high marsh areas have subsided into low marsh or even open water, but that open water may only be 6 to, maybe 6 inches to a foot deep. And that, again, poses some issues. So we worked for quite a long time developing methods, and then started to implement those methods, looking at specifically at these living shorelines approaches. We've had a number of students over this period dating back to 2006 that have put time and effort into this. I'm going to acknowledge them a little bit later, because without their work, we couldn't have, we couldn't be doing what we're doing now. And then we've been very fortunate to get support through a number of local agencies, and that is instrumental in terms of being able to get the supplies and the equipment and be able to get out on the sites when we need to. All right, Haley, next slide. Thank you, thank you. So as Haley said, this is, we're looking at GBF program sites primarily. As she alluded to, many years ago, more than 15 years ago, I ran the program for the Galveston Bay Foundation. And so some of the sites we're studying are actually sites that I was out on at one time, actually doing the design and the construction and working with the landowners. And so again, we developed this partnership. We were able to secure funding. Our funding comes through the Texas Coastal Management Program, and we have new funding starting next year from the Galveston Bay Estuary Program. So again, much thanks to those agencies for providing their support. As Haley pointed out, our sites are they're upper, mid, and lower bay. And so part of our approach here was Galveston Bay is the largest bay in Texas, and the conditions in the bay can really vary from one end to the other. So the upper part of the bay is very highly influenced by the Trinity River, the San Jacinto River. Conditions tend to be more fresh than saline. And so the type of plants, the type of animals that you get there, in some instances can be significantly different than what you might find in the lower bay that is far more influenced by the Gulf of Mexico. And then, as you saw, we have one site that is in a tidal tributary. So Houston, the Bayou City, we have a high degree of small tidal tributaries that encircle the entire bay. And a lot of our private lands and some of our most important fisheries habitats exist in those tidal tributaries. So we definitely wanted to have that represented there. And then in terms of what we wanted to look at, I like whole community studies, and I also see a lot of value in studies that look at biologic communities over time, understanding and recognizing that these communities can change depending on conditions, and a snapshot may not provide you with an adequate understanding of what's actually happening on that site. So, you know, when a site is restored in these low Spartina, predominantly Spartina marshes, the literature suggests that, you know, from the time of restoration to the time that it reaches its peak ecologic diversity can be as long as 25 years. So, you know, going out on a handful of days one summer may not give you a good understanding of what exactly is going on in that site. Okay, next slide. So our objective overall is to investigate the biologic efficacy and both the biotic and abiotic stressors of these small scale restored shoreline habitats. And we want to compare them to not only natural unrestored wetlands in those same water bodies as a control, but we also want to compare them to traditionally armored sites. As Haley pointed out, with private lands, and even in some cases with public lands, in terms of the regulatory hurdles, in terms of cost hurdles, time, oftentimes it's easier, faster, cheaper to install a seawall or a bulkhead of some sort, as opposed to these living shoreline sites, which are a little more involved, in terms of setting up site conditions that are going to be effective over time. So we definitely wanted to include that in our data collection. Really, again, the need here is that much of the existing shoreline and aquatic habitat in Texas is privately owned, and coupled to that, it's relatively small scale relative to publicly owned sites, which, in some cases can have hundreds or even thousands of acres of opportunity for restoration. These sites, again, can be very, very small. There's not a lot of data out there either. So when you examine the literature, a lot of the literature is targeted at these big public restoration sites. And that makes sense in terms of access, in terms of interest in being able to fund those. There's value in having that data. In fact, we utilize some of that data in our comparisons. But having data that is specific to these smaller scale sites, and especially in areas that you might not otherwise find a public restoration opportunity, gives us a whole nother line of understanding of how these sites work over time. So we need the data. We need the data for the purpose of assessment, for the purpose of support to adaptively manage these treatments so that we can correct problems as they occur, but also so that we can improve our restoration efforts as we move forward. So we're working off of the hypothesis that living shorelines are going to exhibit similar ecologic and shoreline stability functions to natural unrestored shoreline habitat, and superior to armored shorelines. Next slide, please. So I'm going to break this out to not get too off track into methods. I'm going to break this out into physical and biologic environment. So in the physical environment, the need here is to document environmental conditions at each sample site, to note conditions that might alter or bias data collection. So for instance, dissolved oxygen, for instance. Dissolved oxygen at these sites in some instances can be very low, and that can have an impact on the presence of things like fish, and can have a long-term impact on community makeup if it happens too often. So we want to take ancillary ancillary water quality measures, ambient air quality measures, and that sort of stuff. We also want to look at stressors like contaminants in the sediments. In these systems, the sediments are where the bulk of the nutrient content, it's a sink for nutrient content that then gets taken up in small organisms, macrobenthic invertebrates, for instance, that then build up into the larger community as a whole. So again, the literature, there's a lot of literature out there about sediment heavy metals and organic contaminants and the impact that can have on those communities. So since these sites are in, the Galveston Bay region, which is very both urban and industrial in some instances, we wanted to take a look at those sediments to determine, okay, what's in these and could that potentially be an issue? And then we also want to document and verify shoreline erosion rates and conduct comparisons of these sites across time to see how they compare against natural shorelines and these armored shorelines. So with our heavy metals, we collect sediments in 2-inch cores. And we're following an EPA method 3050B for digesting the sediments. So you're essentially doing an extraction process, and then you run those extracted liquid on an ICP. U of H Clear Lake is our partner that's been doing this component of that work. For organics, we are using, again, an extraction method using the Quetcher's method, if you're familiar with that. Quick, easy, cheap, effective, rugged, and safe. Gotta love a good acronym. So we're using the Quetcher's method for the extraction process, and then again we're analyzing the liquid in a GCMS here at Lee College. And then for shoreline stability, of course, there's a lot of data out there that's available publicly and privately from satellite imagery. But we wanted to try and get image specific to our sites that was a little higher resolution. And so we are using drones to fly the sites. And so we have drones that are equipped with visible spectrum color cameras, and then also fluorothermal cameras. And so we're flying a programmed pattern over the sites that guarantees we're getting good image overlap, for instance, and allows us to get multiple images of any site. And we can do it on days that we want to be out there where the conditions are best. We're also putting ground control points out on these sites, and the ground control points allow us to establish accurate location and elevation, because elevation is really important in an area that is still undergoing some subsidence. So, and then we're analyzing that via ArcView Pro. Again, we're doing that here at Lee College with some assistance from Black Cat GIS. So that's been really exciting. Okay, next slide, please. So for the biologic environment, here the need is to really characterize the biology of these sites, because what we're doing with these restorations is more than simply hardening the shoreline in an effort to prevent erosion. We want the added benefit of creating and or restoring, in most instances, aquatic habitat that was present at one time that suffered from a combination of shoreline subsidence and then erosion of the sort that Haley showed you earlier. So we want to characterize the plant community. We want to look at the use of that plant community by fish species. We want to look at the macrobenthic community, so the small invertebrates that live primarily on or in the sediments. And then we're also interested in the bacterial communities, the microbial communities in these sediments as well, because of the important functions they have in terms of nitrification, for instance, and nutrient cycling. Next slide, please. So with the plant community in these sites, we're largely following, to the extent we can, we're trying to follow processes that are comparable to processes that have been applied by other researchers at other sites. In this instance, again, these sites are very small. So I expect some of you are probably chuckling, although I can't hear you when I say that we're using 10-meter transects. That's right, 10 meters with 3 1-meter plots, origin center and terminus. And then within those 1-meter plots, we're doing a variety of different metrics. So we're doing stem counts, which allow us to calculate density, diversity, importance values, relative coverage, productivity. With productivity, we are using 2 different approaches. So one thing we're doing is we're looking at chlorophyll production. So we're using a chlorophyll meter in order to do that. And then we're also looking at specifically at Spartina biomass. So with these restoration sites, like many, we are using an anchor species. In this instance, Spartina alterniflora, Gulf cordgrass, is pretty ubiquitous across the Bay system. It is a facultative species, so it tolerates very saline environments of the Gulf Coast, and also thrives in freshwater areas as well. So when we construct these sites, Spartina is typically our go-to plant in terms of what we're going to sprig the sites with. For the Nekton community, this has been probably one of the bigger challenges, is trying to figure out how to collect statistically measurable, comparable data. So trying to pull, again, I mentioned these are subsided sites, and a lot of times there are still a lot of snags from trees and other plants that were, where that was an upland site at one time, they're still underwater. And so trying to pull a seine, for instance, is virtually impossible. Some of the sites are saline, so you can't electroshock. So at a Ray conference, actually, I think it was like 2006, we were troubleshooting this a little bit as we were trying to figure out our methodology. And my students and I had seen a couple of presentations where these researchers had applied pike nets at some larger sites up in the Northeast. And so again, pike nets in these waters, because they're so shallow, aren't really a very viable approach in some instances. So, but we did think, well, in many instances, the fish that are accessing these sites are, in most cases, pelagic species that are coming in so they're coming in from the Gulf of Mexico, they're laying eggs, they're feeding, and then so a lot of the species are small schooling species like Gulf menhaden, for instance, Gulf killifish. And there are some larger species as well, but we thought, well, the most access is probably going to be through those smaller species. So we decided to employ these small 18 by 10 little collapsible minnow traps you can buy from Bass Pro Shop. In fact, I think that's where we got them from. And so we put them out. They stay in place over 24 hours so we can capture the diurnal cycle. We go out, we collect, and in most instances, we're returning most everything back into the water live. In some instances, if we need to do identification so we can't do it in the field, then we'll bring them back. We're looking at benthic macroinvertebrates, again, using 2-inch stainless cores. And we're analyzing for species richness, diversity, total abundance. And if you're familiar with it, some of my work has been up in, so I'm an aquatic toxicologist by training. And so some of my work's been up in the Houston Ship Channel working in Superfund sites. And so there are metrics like EMAP that are used to make comparisons looking at sensitive organisms or specific communities of organisms or populations of organisms. So we're going to use that approach as well. And then the sediment microbiome, again, the interest here is, looking again at the whole community function. So we're using, I don't know if you're familiar with the next gen sequencing system, but it looks at the 16S RNA sequences. And then we're working out the phylogenetic taxons using the bioinformatics that go with that. Okay, next slide, please. All right, so what are we finding here? I have to preface this. Like many of my colleagues across the country, many of us had significant setbacks as a result of COVID. So we were set back nearly 2 years in our efforts to get out here and do this. And we are still actively collecting data. In fact, I was out the end of last week, and I'm going to be out the beginning of next week. We're still doing active nekton collecting, for instance. We're still analyzing samples in the lab. So what I'm going to give you today is very preliminary, but hopefully it'll give you a little bit of a picture of what we're finding. I would certainly welcome, feedback and insight, recognizing the expertise of many of the attendees I see are on the webinar today. So do feel free to reach out if you have suggestions or questions. Okay, let's go to the next slide here. All right, so again, I can't present everything because we're still doing some analysis. One of the areas because we're still collecting data, we haven't done a lot of significant statistics. Come on, that's a good dad joke. I hear you all laughing. So we haven't done a lot of comparative statistics yet. So I'll share that where we have, but for the most part, this is, just to give you a picture of the kind of things we're finding. So with heavy metals, again, we ran these on the ICP. You can see here that looking at heavy metals, we do have some statistically significant differences between the sites and between treatments, but it's fairly specific to what the metals are. So for instance, you can see in the top graph there that, total heavy metals in our Armand Bayou samples across the treatments are all higher than either Trinity Bay or West Galveston Bay. That's not too surprising given the nature of that particular tidal tributary that drains an area that is both very urbanized, and the headwaters of that particular bayou are near an industrial part of Houston as well. So that we're finding some heavy metals in there isn't particularly surprising, especially compared to the literature. The important thing is, so I show Armand Bayou down there to the bottom. The only metal that we found that has exceeded the Long and Morgan ERM standard is for copper in our Living Shoreline site at Armand Bayou. And even there, you can look at the bottom there to the right. The standard is 34 parts per million. Our finding was 34.8. So we're not talking about anything that would be particularly concerning to me as a ecologist and toxicologist. So when you look at this data, even though you're seeing some stuff here, and there are some significant differences in terms of the amounts between the sites, in the bigger picture, these are not concerning values. So we're happy about that. That was uniform across the sites that we're seeing. Next slide, please. So with organics, we had a similar result. So again, we ran this through the GCMS. In this instance, we're looking at, we're using a method, it's the EPA 8081, which looks at 20 organochlorine pesticides. Our thinking being that in these areas, so for instance, again, looking at Armand Bayou is in a very urban residential area. It would not be surprising to, find some very low levels of organic pesticides there. Same thing over in the Trinity Bay system where there's a lot of agriculture. There's some potential there. So we wanted to examine that. You can see our process here. I won't go through that in a lot of depth. Rather, I will point out that our detection limit here is good. We're able to detect down to 0.02 parts per million. And we did have some hits. They were very, very few and far between and very small. None of them exceeded None of them exceeded any of the protocol that we've been examining so far in terms of health and safety standards, which in some instances for some of these pesticides, especially legacy pesticides, are more geared towards human health in many instances than ecologic health. So we're still working through that, but again, the values are all very low. And what we have suggests that there is no difference among the different site treatments. In most instances, I will tell you that we found no values in the sample sediments. Initially, we were kind of surprised, and we thought, well, we go back and look at this again. So, we run controls when we do these. Same with the heavy metals. We run controls so that we can calibrate the equipment, ensure that it is detecting what's in the sediments. And so this is a good finding. We're really happy with this, and it's consistent with what we found with the metals as well. Okay, next slide. For shoreline stability, as I said, we're out there. So we wanted to fly low altitude. So we're flying under 400 feet. In some instances, we're flying as low as 30 feet off the ground. This has probably been one of the most interesting parts of the study for me personally, because I'm the one that's actually flying the drones. And that was quite a learning curve for me. There's a license that goes along with being able to operate the drones commercially. There's a lot to learn in terms of how to capture the imagery and to get good imagery so that it can be well utilized in the GIS. So we've had a couple of partners work with us along the way to get up and running. And so you can kind of see what's going on here. One of the things you have to be really concerned about, especially in an environment like Houston, is airspace. So that was where the licensing, I think, is really helpful. So anybody who's watching this presentation and you're thinking, oh, this would be a neat thing to employ, do look into the federal licensing so that you're aware of what the requirements are for you, whether you might fit any of the exclusions or whether you would need to have that license. I personally recommend the schools, because there's a lot about airspace I didn't know. And so I have this one particular picture there off to the lower right, because we live in areas here in Houston where we have commercial airports that overlap with military airspace. And then there are a number of restricted areas where you have to have special permissions to get in and fly the drone at all. So navigating all of that has certainly been exciting. So when we go out there, of course, before we do anything, we have to set up ground control points for the sites, and that ensures that we're able to locate the site accurately in the GIS, and also ensures that we're getting good elevations. Again, this is an area that has undergone significant levels of subsidence since the 1950s. And some areas, while subsidence has greatly slowed down in some areas of Houston, it has accelerated in others. And so there is still some subsidence occurring. So we want to definitely monitor that as well. So to do this, we're using these little, this is the funniest thing. Rather than there are a lot of commercial ground control points out there that you can find, but I just went to the dollar store and got some Frisbees, screwed them to the top of a 1 meter PVC post. And so there's a tab at the bottom of that pole, so you can see them sticking out of the marsh there. So those are 1 meter off of the sediment line, and each Frisbee has a letter painted on the top of it, so we can recognize it from the air. The students then go out with a RTK GPS, and they're locating those, and then we'll actually fly the drone at that part. All right, so you can go ahead and next slide. Haley, thank you for doing this, by the way. All right, so for the next step, then we take those images, and usually we're running 2 or maybe even 3 flights at a site for both the drone that has the true color imagery on it, and then we have a different drone that has the FTIR. I'm hopeful with some of the new equipment we've got, I'm going to be able to do both with one drone at the same time. So that'll be extra. That'll be extra good. So here you're looking at one of our sites in Clear Lake Park. So we fly all those because in many instances, when you're trying to fly a drone, these drones, even our biggest one is, these are commercial grade drones. I'm sorry, consumer grade drones. So in this instance, this was flown with a little Phantom 4, a DJI model drone. And the drones have very good stability controls. The software is really amazing. The flight controls are really amazing. But nonetheless, when you're working at coastal sites that might have winds at 10 to 15 miles an hour with gusts to 25, that can create some challenges in getting consistently good imagery, even across small sites like these as you're flying the drone along its pattern. So we're pulling the best images, putting them together in a mosaic, locating the ground control points. So you can see here, the red dots are where those ground control points are. And then, so we can pull those images into a single raster. And then the students are working with our GIS specialist, Amanda Hackney, many of you on this call probably recognize, is working with them then to cluster these. And then we're classifying them based on color analysis. So we are still working through this. I don't have any real meaningful imagery to show you beyond this at this stage. This has been the slowest process, but I will show you something we're working on. If we go to the next slide. So here, I mentioned the color infrared or FLIR imagery, and so I thought I'd show you what that looked like. So here you're looking at one of our sites on Armand Bayou, and to the right, so this is a visible spectrum image, right, natural color, and you can see our ground control, 3 of our ground control points in there. And so this is what that marsh looks like. And so we set the ground control points up essentially where our transect is, so that the data we're getting from our imagery aligns with the data we're getting from the transects themselves, from the traps, all of that stuff, ultimately so that we can do the statistical analysis on it. Well, one of the things we're kind of interested in is whether the color infrared might give us a different set of data that we could apply to these sites that might be just give us a whole different level of information. So if you're not familiar with color infrared, instead of measuring reflected visible spectrum light energy off of whatever the surface is, so you're seeing the colors as your eyes would see them, rather what the FLIR image or what the FLIR camera does is it measures the temperature off of that surface or set of surfaces. So in the right image, you're seeing the FLIR imagery, and up in the top left corner, you're actually getting the temperature analysis for the minimum and maximum temperatures that the camera is seeing at the time. So in some instance, even our coolest temperatures, which are represented in blue in that image, are just under 91 degrees Fahrenheit. The orange areas, the bright orange image areas of that image are reflecting at nearly 120 degrees Fahrenheit. So I tried to align these images so that they're at the same scale, so we can make some comparisons here. And in the FLIR image, I'm actually captured at the bottom down there. That was just kind of happenstance, so you got a sense of scale here. So you can see where our ground control points are, and with those ground control points in the visible spectrum image, that marsh looks fairly thick, and it is in terms of the shoot coverage that we measured. And you'll see when I present the plant data here in a few minutes, you'll get a sense of that. But with the FLIR imagery, what we're seeing there by the color analysis, it's allowing us to pull out the areas that are higher marsh and see the areas that are inundated, and we can get a better sense of where the inundation stops. So you can see in that FLIR imagery down at the bottom where I'm standing there, that line, that green line that I'm standing right next that then transitions to blue is where it transitions from upland to high marsh. And that is a fairly steep shoreline in that bayou. And it's characteristic of most shorelines in these tidal tributaries, where it drops off pretty rapidly. And in the visible spectrum imagery, right, to the left of that, you can see that there's a little bit of a, well, you can actually see that St. Augustine grass there that we're looking at on that ridge, and it's mowed. I'm standing right next to it in the FLIR imagery, and then you can see how those plants transition as you go down slope. So the interesting thing we're looking at is to try and determine, okay, how can we utilize this? In some instances, CR imagery has been used to identify individual species of plants. Trees, for instance, in forested areas put off different heat signatures, and you can actually speciate trees based on their heat signatures. In a marsh where you have as much, where the plants are so intertwined with each other, I think it's doubtful we're going to be able to do that. But it can give us, I think, it will provide some value in monitoring the marshes over time, where we can see where marshes are opening up. So for instance, here in a few minutes, I'll talk a little bit about some of our sites where what I mean by opening up is as the sites continue to change, in some instances, there's still subsidence going on. And so the density of the plants is decreasing, and there's more water in those sites, and it's becoming more inundated. Well, if, implementing a field visit that's 2 to 3 days where we're going out, we're running these transects, and we're setting traps, and we're taking samples, and then doing all the lab work that goes along with it to get back to characterize these sites, is probably not something that most entities are going to want or even maybe be able to afford to do on a yearly basis, right? In fact, when you look at marsh construction, the requirements from granting entities in many instances for monitoring on the back end of these efforts is, pretty simple. It's, let's look at how many plants, how much spartina is there, what's the relative coverage, and that may be it in many instances. So what we're hoping is that utilizing this technology, these two things side by side, along with the data that we then collect maybe once every 3 to 5 years, might provide a streamlined, more cost-effective means of monitoring a site over time. And with both the true color and the color infrared imagery, along with the ground truthing that we're doing through our site visits, we might be able to collect data to monitor these sites on a much more frequent basis, so we can get a sense of how quickly they're changing between our big data collection events, that can provide a lot of insight in terms of how these sites perform over time. So I'm really excited about this, you can see, because I'm talking about it so long. So.
Yeah, Jim, I am terribly sorry to have to do this, because this is fascinating, and there is such an interest in this topic, but we are at the top of the hour.
Gotcha.
If I can give you a minute to sort of wrap up. I know you've got a couple of slides you wanted to get through,
Sure.
but
Sure.
we do need to wrap it up at this point.
I am really sorry. I think I went too long.
This is absolutely fascinating stuff. And I know that our audience is very, very interested in this, but we are at the top of the hour at this point.
Well, we'll have the whole slide so they can see the data, but Haley, if you kind of poke on this, yeah, let me talk about this, and then we'll flip to the last couple of slides, and we'll get it wrapped up. I apologize again. So here we're looking at plant community data, and with this data, what you're seeing here is let's look at Trinity Bay, for instance. So the leftmost data. So we've got data that looks at 2 different years, because we did a major data collection event in 2018 as well. And you can see the reference site there in terms of the importance of the species. So in terms of, what their coverage is, their density, all that sort of stuff has stayed relatively stable. If you look directly to the right of that, you can see that the not only are the what species are present changing, but their relative importance throughout that is changing as well. So that tells me that site, that Living Shoreline site, is still in transition, that we're still moving it, that it's still moving towards its ecologic peak. And you see that in the data at the other sites as well. So that would reflect the relative young age of many of these sites. let's move forward here, Haley. You'll see that also in the plant community data. One nice thing, one of the things we're doing when we look at biomass is we are looking at both root and shoot data, because the roots have such an important function in how they moderate and change the sediments. So that makes the sites more resilient over time, the effect that they have. So that data is there, and you can take a look at that. We are seeing some declines. Some of that might be related to some of the environmental conditions we've suffered over the last couple of years, including this big freeze we had back in February. So we're looking at that as well. Let's move forward another couple. With the Necton community, data here to us, it's still kind of all over the map. That tends, I think, to reflect the transient nature of many of the fish species we have. So we're still looking at that. I'll leave that data for you to look at and check out. Let's go forward a little bit. With the macrobenthic community, one of the neat things we're seeing is that if you look at this data, our species diversity tells one story, that we have pretty broad diversity and pretty broad species richness that's reflected in all of these sites in Arm and Bayou. So this is where we have the most data complete at this point. But if you look at the abundance, you can see that the abundance follows a very distinct trend. Highest at the reference site, lowest at the armored site, with the Living Shoreline site being in between. Again, suggesting that the sites are still undergoing their ecologic successional processes. And that's something that we want to see as we move forward. How quickly does that occur? How successful are they getting there? Okay, let's flip forward again here, just another minute or two. Our biologic community data seems to suggest that there might be some issues from erosion, particularly at our reference sites. So that's something we're monitoring as well. We can go forward again. So our next steps, we are continuing to monitor. Our next big field work is going to be in the spring and summer of next year. We are looking to add some new metrics to this, including microplastics, because of the nature of the sites, looking at how microplastics, we find a lot of debris in some of the sites. And so how are those sites affecting, are they breaking down? Are those sequestering to the sediments? Okay, and next slide. So I want to acknowledge our research team here and faculty who have worked really hard, our partners. And then last slide, please. Also our partners, again, very much thanks to all of these folks who have done everything from provide really important funding, the coastal management program, the estuary program, but also technical support in terms of providing facilities, expertise, and that sort of thing. So thank you again. I apologize we ran over a little bit. My fault there. That's not unusual for me. And so we thank you for hosting.
A huge, huge thank you to both of you. This is absolutely fascinating. There is just a ton of information in here. And I know we've got a lot of interest and a lot of questions. We are over time. So I'm going to offer you one question, and then I will say to those of you who did drop some questions in, thank you. I apologize we didn't have time to get all of them, but do feel free to follow up. You can get in touch with me or directly in touch with the presenters. And the one question I'll put forth is, the data collection and studies seem to focus on marsh success, not the success of or comparison of living shorelines versus traditional armoring. Are there monitoring metrics that you would recommend specific to living shorelines that show the benefit or the difference compared to traditional armoring?
Haley, you mean so
I could go to either one of you. You have metrics in mind. I know we have a strong community of practice with folks working in living shorelines around the country.
Sure.
And monitoring efforts. There's never enough budget for it. And so any tips about how to methods or ways that on a thin budget that they could monitor after a living shoreline has gone in?
Well, I don't mind.
Oh, go ahead, Haley, go ahead.
I'll kick it off. I see that's from Kristen Hames, who's with the state here, and we work together quite frequently. Great question, Kristen. So this study does look at 3 different sites in each part of the bay. So Jim is comparing all of these metrics that he talked about on a living shoreline project, on a reference marsh, and on a bulkheaded or armored site. So yes, that is all being compared to kind of address that first component. As far as monitoring metrics, I'll maybe kick that part over to Jim.
So, so Haley's right. We're looking at all 3 sites, and the idea is that with the comprehensive nature of the data, there's a lot of different comparisons we can make. So we can compare between living shoreline sites. We could look at construction methods if we had enough data. We can compare. So if I understood the interest is in comparing the living shorelines to the armored sites, and that is specifically part of what we're doing. So all of the metrics that we take at one site, we take at all of the sites. So of course, there are no plants at the armored sites to look at, but by measuring the benthic macroinvertebrate communities, the nekton communities, looking at monitoring erosion as well. Haley showed you pictures early in the presentation about the failure rate of these armored sites. In fact, one of our sites up in Trinity Bay, we've been slowly watching the bulkhead, this vinyl sheet pile bulkhead, fail over the last 10 years. And so these metrics should be able to bear that out over time.
All right. With that, I'm afraid we will have to wrap it up. Thank you to those of you who were able to hang with us for the extra few minutes we've run over here. This recording will be available. Give us a couple of days to get it processed, and we'll have it up on our YouTube channel. There will be a link from our website. And I can ask if you as presenters are willing, I can make the slides available as well. There's a ton of great data in there. I know you may have publication issues there, but I know that there are definitely folks who will be interested in that. So again, give us a few days and we'll have the recording available. Thank you to everyone who was able to stay with us today. And a huge thank you to Haley and to Jim for all of this great information. Thank you so much, everyone.